Okay, guys, here is our content for today. So today we're going to start just by looking at our task verbs, and we're gonna look at these three task verbs. You've seen them before. Okay, identify, describe, and explain. You're gonna see a couple today. We will kind of review these task verbs as we go, but you guys saw all this vocabulary yesterday, so hopefully you're feeling good about these. Remember, identify is 1 in terms of detail, describe is 2, and explain is 3. Okay. Identify, you do not need to write anything but 1, maybe 2 words. Okay, do not write more than you need to. Okay, that's identify. Describe, requiring relevant characteristics. Okay, and these relevant characteristics of the specified topic can usually be answered in a sentence. You need to use your actual passage to figure out what it is that you need to describe. Okay. But this is kind of a 2 in terms of detail. The explain will require a little bit more and usually is going to require evidence, okay, and or reasoning to support or qualify a claim. So you really want to make sure that you are going into a little bit more detail when you're asked to explain, especially when that refers to a trend, okay? So moving into experimental design, and data analysis. Whoopsies. So I want you guys to be able to look at an experimental design and 1, be able to analyze the data from it, and 2, be able to figure out what the parts are. And this is because these are the questions that are there's going to be 1 question on your AP test that will talk about experimental design. It'll give you a new experimental design, and you need to be able to break it down. You need to be able to find those different parts, and you need to be able to analyze the data from it. Okay, these are things that you guys have done. I know that you can do this. We're going to do some really easy practice to start, and then we're going to build up to some harder questions from past AP tests. So let's say that you have an experiment, right, that is testing what we see in this diagram, okay? They will give you more context than this, okay? But we can see that this is probably the purpose of this experiment, okay, has to do with our dependent variable, which is going to be the data collected, the independent variable, which is our what's being manipulated, in our control group, which is used for comparison, okay? So they tell us our independent variable is the amount of water, our dependent variable is the fraction of the seeds that sprout, so how many germinate. So really our experimental and our control groups they show here, right, are no water or some water, right? So the presence of water is the amount of water is our independent variable, and our experimental control groups are shown here. Okay, the control group is used as a comparison to see what happens when we do add water. Okay, and then our experimental or independent and dependent variables help us figure out what the purpose is. Okay, so the purpose is to figure out what is the effect of amount of water on the fraction of seeds that sprout, the germinating seeds. Okay, and we can clearly see then when we're looking at data analysis, we can clearly see, right, that there are 9 out of 10 seeds that sprout in this experimental group where water was added, and there are 0 out of 10 seeds sprouted in water, right? So we know that that when you add water, it will increase, okay, the seeds that sprout. This is a very simple setup, but I want you guys to continue to go back to these more simple setups when you're given really complex experimental design setups, okay? The reason I included this is because remember that we also are going to have some things that are held constant so that we know that anything we see in our data was due to a change in the independent variable and not anything else, right? Make sure we have the same seeds, we make sure we have the same soil, right? Identical pots like it says here. And this is going to be, we saw this yesterday in question B in FRQ 2. They asked you what the purpose was of changing the amount of organisms that were put in group 2. And the reason for that was to keep it the same as that first group, okay? Cool. let's very simple way that we could look at results here. Nope, that's not what I want to do. Right? This is a really simple way that we could see results right here. And I think that this is probably not going to be the way that you guys see results. Okay? You will see something. So we could also see results that look like this, right? This is like this, right? A graph or a table, which you guys have seen. So I want you to take a second and yes, Germany's C's versus number of days, right? This is showing something similar, our independent to the last experiment, but our independent variable here is going to be what we see up here in the legend. So this is usually going to be some sort of the IV, the independent variable. So we're changing concentration of NaCl, salt, in this water and seeing how it affects that, right? So we see, right, that there's nothing for these last two, and that for 0, which would be pure water, we have the highest amount of germinating seeds over time. Okay. I want you guys to do something similar. Okay, we have the same idea. So concentration of polluted water, not salt, but polluted water here. Okay, I want you to describe, so remember this task verb, describe what occurs as the concentration, this should say, as the concentration of polluted water increases. So describe what occurs. Describe essentially what the trend is that is most important. Please write this in a complete sentence. Okay. Next, okay, we have our check for understanding. So this is that part. Last part was a little bit of a quick check for understanding, but this part is going to be a little bit tougher. It's going to have, this is actually from an AP test. Okay, so I want us to take a look at this. So the common bedbug, okay, is a species of insect that becomes increasingly resistant to insecticides. Bedbugs possess several genes suspected of contributing to the resistance, including these 2 genes. Okay, 2 genes, P450 and ABC, as well as CPS. Okay, to investigate the role of these genes in insecticide resistance, researchers deleted 1 or more of these genes in different strains of bedbugs. Okay, so they're deleting these genes in different strains of bedbugs, as indicated in figure 1. And the treated strains with the insecticide, beta-cyfluthrin, each strain was genetically identical except for the deleted genes and was equally fit in the absence of beta-cyfluthrin. The percent survival of each strain following beta-cyfluthrin treatment is shown in figure 1. Okay, we know that if this is what's being collected, that this is our dependent variable, is our percent survival. So if you were graphing this, for example, right, and they have this here, it's going to be here. So your dependent variable is almost always going to be over here. Okay, these groups, okay, these strains that have different deleted genes, okay, are shown here. So each of these groups, right, correlate with the data above this. Okay, and you also have, okay, what in each strain, which genes were deleted. So all I want you to do for this, and this had another part to this question on the real exam, but all I want you guys to do with this is identify the control strain in the experiment. Okay, and I want you to use the means and the confidence intervals in figure 1 to justify the claim that ABC8 is effective, so it's good at providing resistance to beta-cyfluthrin. So I want you guys to try this one out. I broke it down a little bit for you guys, not completely, okay? But I do want to note that this information is going to be important here. Okay, the percent survival of 5 strains. Okay, the percent survival with beta-cyfluthrin. The positive indicates that the gene is present, and the negative is that it's deleted. Okay. One last thing before you guys get a chance to actually try this is I would like for you guys to go ahead and actually look at your standard error bars. So remember these standard error bars, okay? If they overlap, meaning these two, for example, overlap, this means that these two groups have no significant difference, okay? And the reason that that is important is because if you are looking at your if you're looking at justifying a claim, right, that it's effective, you need to bring in evidence here and you need to use those confidence intervals, right? These two groups, for example, there is no significant difference. But if you look at maybe two other groups, like, let's see, let's get rid of some of this. If you're looking at two other groups, okay, like, let's look at groups. Sorry, guys. Let's look at this group, group 2, and group 5, right? Group 2 does not overlap with group 5 standard error bars at all, right? So we would say group 2 and group 5 are significantly different according to the data. My handwriting has gotten even better on this app, guys. Okay, go ahead and try this on your own. Okay, so let's go over this. Your control strain was strain 1, okay? All you needed to write was strain 1. You did not need to justify, okay? But if you did justify, right, strain 1 has all of the genes, so this is our point of comparison because this is kind of like the quote-unquote normal because they normally would have these genes, right, or genes. So we're manipulating them to not have the genes in these experimental groups, okay? So this is our control. We know that now. Cool. Our B, okay, and again, sorry, just to go back to control. If they just ask you to identify, great. If they ask you to justify or to explain the purpose of the control or something like that, make sure that you write that it is a point of comparison, right? It's to make sure that we can compare these experimental groups to some sort of baseline, okay, which is 1. Good. And then justifying the claim that ABC is effective at providing resistance. Well, ABC is here. Okay. And the fact that we see that whenever we have ABC, okay, or when we have our control strain, those error bars from the control do not overlap with strain 3 where it's deleted. Okay, so if you see here and here, okay, strain 1 and strain 3 do not overlap, which means there is a significant difference, right? Between the control and group 3, or where ABC is deleted, right? That's what group 3 is, strain 3. Okay. You could also say that the mean percent survival of strain 3 or the ABC deletion falls outside the 95% confidence interval, or that there is a statistically significant difference between strain 1, the control, and strain 3, which is here. Okay. The reason that we can't compare it with 5, which is also where it's deleted, is because we also have P450 deleted. So this is the only strain that's showing the ABC. So this difference tells us, right, that ABC is effective at providing this resistance, because when it is not there, right, we see that it's very different from the control when it is there, right, and we have everything we need. So I do just want to make sure that we're understanding that when we're asked to justify in this case, justify a claim, okay, we have to bring in the specific data from this table. Okay. Great.